Good morning, everyone. We're going to try and get you through this Monday morning. Thank you for joining us. I'm Steve.
And I'm Scott.
And together we're Backyard Musings, the science and technology channel.
Yeah, and it's early. And today we're talking about our I don't know if it's our favorite disease. That's probably not the best way to put it, but it's a disease that we like to talk about when they're coming up with reasons to get rid of it. And that would be Alzheimer's disease. So let's get into it.
Yes, sir. Scientists have identified an experimental molecule that appears to restore some of the brain's natural defenses against Alzheimer's disease by helping immune cells once again contain the toxic protein deposits linked to the condition. The findings, published in the journal Cell Death and Disease on April 27, 2026, showed that the molecule called OLE reduced harmful beta-amyloid plaques and improved memory in animal studies. Would you like to know more?
Okay, one of the hallmarks of Alzheimer's disease is the buildup of beta-amyloid plaques in the brain. Normally specialized immune cells called microglia, and we've talked about that before when we've talked about Alzheimer's before, help clear away these toxic protein deposits. But as Alzheimer's progresses, microglia gradually lose much of their ability to protect the brain, allowing plaques to accumulate, continue, or contribute to damage of nearby neurons.
The research discovered that OLE, a molecule derived from the PM20D1 gene, can help shift these immune cells back into a more protective state instead of becoming dysfunctional. Treated microglia moved toward beta-amyloid plaques and surrounded them, creating a protective barrier that limited the plaques' contact with neurons. This reduced both the size of the plaques and their harmful effects on brain tissue.
Okay, quote here. One of the most significant findings is that we have identified a molecule capable of restoring microglia's protective function. And that's from Sanchez Mut. Probably German, I don't know. Going on with the quote, in Alzheimer's disease, these cells become progressively impaired. Our results suggest that this process can be reversed, pointing to new therapeutic and research avenues to counteract the disease. Excellent. And that's according to a researcher who leads the functional epigenomics of aging and Alzheimer's disease laboratory at, I don't know what this is, INCSICUMH.
Yeah, some university.
University of Munich, maybe? I don't know.
Yeah, maybe. To evaluate the effects of OLE, the team tested the molecule in several experimental models. The first experiments used genetically modified worms, C. elegans, that produce beta-amyloid, allowing researchers to quickly study the protein's toxic effects. Worms treated with OLE accumulated fewer protein aggregates and showed improved movement, suggesting the molecule reduced disease-related damage.
Okay, I don't know if we've ever done one of these where they've used worms, right?
Right.
Interesting. The researchers also treated mouse models of Alzheimer's disease with OLE for 3 months. After treatment, the mice performed better on memory tests and had fewer beta-amyloid plaques in their brains. Okay, that's positive.
To better understand how OLE works, researchers examined the activity of thousands of individual cells in the brain. Their analysis revealed that microglia were the cells most strongly affected by the treatment. After receiving OLE, these immune cells activated pathways involved in clearing beta-amyloid and regained their ability to migrate toward plaques and surround them.
Okay, another quote. Single cell analysis allowed us to determine the microglia were the cells that responded most strongly to the treatment. And that's according to Victoria Pozzi, first author of the study. Going on with a quote, from there we observed that the compound helped these cells move toward beta-amyloid plaques and better contain the damage associated with the disease.
Additional laboratory experiments supported these findings. In cell cultures, OLE-treated microglia became more effective at moving toward beta-amyloid deposits and promoting their removal. in separate neuronal cell cultures exposed to Alzheimer's-like stress. The treatment also increased neuron survival, suggesting the molecule may directly protect nerve cells as well.
The researchers say the findings highlight OLE's potential as the basis for future Alzheimer's therapies. The work is protected by 2 European patents, including one owned by the CSIC, reflecting its possible translational and therapeutic value. So I maybe I'm missing the point here, but I got a problem with this patent thing. So, and I'm not a patent guy, but that usually means that this is our research, our deal, right? You can't really, unless you want to pay for it, you can't use it, right?
Yeah.
We're more into the collaboration stuff, right? Let's just get this ugly disease off the table. And, but I don't know. Maybe there's some monetary deals going on here. Who knows? But
there's 2 kinds of doctors, Scott. One's in it for the money, and one is in it to improve health.
Yeah. And if I'm reading this right, it sounds like these guys are maybe in it for the bucks. But I don't know.
Yeah.
They can cure it, whatever, you know. But you'd think that they'd be wanting to just be cooperative and get everybody involved. You know, I would think the money would probably pour in more for their studies than they would be if they're or that they've got the, you know, God's gift here. So, but anyway, we'll see.
This is good news, though. Let's see where it goes.
Good, definitely good news.
All right, folks. Thanks for joining us. We'll catch you in the afternoon.
Take care, everyone. Have a good morning.